The LYriCAN project is an integrated site for innovative research in oncology. It is supported by the French National Cancer Institute, the INCA, since 2011. It gathers labs of excellence around basic, translational and clinical research in oncology. There are three ambitions for the LYriCAN, which are actually identified as three integrated research projects. The first goal is to better understand the mechanism of resistance, primary and secondary resistance to treatment. And the angle of investigation is a precise characterization of the epigenetics of the tumor, on top of the genomics of the tumor that we have been working on and still are working on in the previous LYriC program. The second one is to identify combinations of treatment. Single agents are not extremely efficient. Combination of treatment should be more efficient in selected patient population. That's a question. And the third one is the identification of better treatment of the macroscopic tumor. Surgery and radiotherapy are not always achievable in some patients, and we want to delineate novel treatment involving new physical strategies, involving mathematics, to better treat a tumor which cannot be treated as we speak. So again, it's always about better understanding what is ongoing in terms of resistance and providing better treatment to the patient.
We strongly believe that beyond the intra-tumor genetic diversity, which comes from the Darwinian process of tumorigenesis, cancer cell plasticity is a major cause of resistance to treatment. So over the last few years, we have developed strategies based on drug screening strategies, aiming to identify small compounds which are able to block the EMT process. And these drugs are expected to hamper the evolutionary potential of cancer cells and to make these cells more sensitive to chemo and immune therapies.
Within the LYriCAN, there are several topics dedicated to hematological malignancies, in particular lymphoma. More specifically, during the LYriC program, we have developed a genomic approach to identify a prognostic score built on 23 genes, helping to identify the patients that will do well with standard therapy from those that will have an adverse outcome. So recently, in the LYriCAN program, we are developing a new approach to identify specifically the genes identified in each individual tumor cells and their microenvironment, using an in situ hybridization process to better understand how immune checkpoint blockers work in the field of Hodgkin disease. With LYriCAN, I believe that we have a fantastic opportunity in Lyon to gather both clinicians for both institutions, Centre Léon Bérard and Hospice Civil de Lyon, to really exchange on a daily basis and try together to solve some of the issues.
Based on the discoveries that we have been doing on this concept of dependent receptor, we have been showing that one specific ligand called netrin-1 was actually upregulated in a large fraction of human cancer. So it was basically the discovery we made during the LYriC, the first LYriC program. And then we developed this antibody that is targeting netrin-1, so this antibody is called NP137. We are doing the phase 1, first-in-man, first-in-class of this monoclonal antibody. And the idea will be that in next steps, the next 2 or 3 years, we will actually try now to combine the netrin antibody plus different types of chemotherapy or actually immune checkpoint inhibitor. So this is the whole goal of this LYriCAN project.
As a nuclear medicine physician, I develop and I organize the network surrounding the development of new traces, new radiopharmaceutical. The IRP3 deals about physical agents to overcome tumor plasticity. We only give macroscopic answers to that questions, but they are all treated with different tools, like we use ultrasound to treat patients with HIFU, either external beam radiotherapy or internal radionuclide therapy. The IRP3 is related to the other IRP through a molecular imaging. We use the biomarkers that are developed by the other IRP, like netrin. We have developed antibodies that will target this netrin, either to proceed imaging as well as doing radionuclide therapy. This is what we call a theranostic procedure.
Equal access to innovation is a real issue in France and probably in the major part of the world. And one of my works is working on this topic and try to identify the real mechanism of inequality and how to resolve this problem for the future. And we would like to develop more and more research programs where the patient is not only a patient but also an actor.
There are so many challenges in oncology as we speak. First one is the integration of patients in the definition of research objectives. The second one is, of course, to identify better which are the nosological entities and the new targets. The third one is, of course, integration, bringing together basic, translational and clinical researchers together to a single goal, which is to bring new service to the patient, is something which is, of course, mandatory.